Dear student, today we are going to study radio immune assay, that is revision class. Here you can see in screen radio immunoassay. Immunoassay. An immunoassay is a test that use antibody and antigen complexes. An antibody antigen complexes is also known as an immunocomplex. Immuno refer as immune response that causes the body to generate antibodies. Assay refer as test. Immunoassay is a test that utilize immunocomplexing when antibody antigen are brought together or combined together. Here we must know some definitions. An antibody is a protein that is produced by the body in response to a invading or foreign substance. Antibodies are produced as a part of body immune response to protect itself. And antigen is the substance that the body is trying to fight off by mounting an immune response. Example, the drug is antigen that bind to the antibody. And immunogen is a substance that elicits immune response. Example, drug protein conjugate. An analyte is anything measured by the laboratory test. The immunoassay testing, the analyte may be either or an antigen. Immunoassay utilize one or more selected antibodies to detect analytes to interest. The analytes being measured may be number one, that are naturally present in a body, such as thyroid hormones. The body produce but are not typically present, such as cancer antigen. Do not naturally occur in a body, such as drug abuse. These are the analytes measure. Now, structure of antibodies. Antibodies are produced by the B lymphocytes or beta lymphocytes. The most common one is immunoglobulin G, that is also known as IgG. IgG is a protein composed of two main structural and functional regions. That is divided into, that is given in a figure. You can see Fab region. It contains the antigen binding site that varies between different antibodies. And FC site or FC region, this region of constant structure within the antibodies. Now, RIA, what is radioimmunoassay? Radioimmunoassay is a elegant technique in analytical chemistry. If substance to be analyzed is in very low quantity, in the order of microgram, nanogram, conventional method like gravimetry and calorimetry methods fail. Radioimmunoassay find extensive application in the assay of many substances which are present in a trace amount in a blood. The amount of AB per tube is kept constant. The amount of antibody per tube keep constant. The amount of antigen added is variable. Different uh, quantity of antigen we are adding. The added antigen will be distributed between a bound B and free F fraction. The distribution is governed by the association constant. This Ka is a assistant constant of antibody. Antibody, this equation is given here. AB plus antibody plus antigen combined antigen antibody complex, and association constant is Ka equal to AB Ag divided by AB and Hg. Competitive binding of radio labeled antigen and unlabeled antigen to a high affinity antibody. The labeled antigen is mixed with antibody at a concentration that saturates the antigen bind sites of the antibody. As the concentration of the unlabeled antigen increase, more labeled antigen will be replaced from the binding site. Here you can see the decrease in amount of radio-labeled antigen bound specific antibody in the presence of test sample is measured to determine the amount of antigen present in a test sample. In the standard condition, amount of labeled antigen bound to the antibody decrease as the amount of unlabeled antigen. Here you can see green is unlabeled antigen and blue is labeled antigen. In diagram you can see you have to draw this diagram in exam when you are explaining radioimmune assay. A reagent used in RIA, radioimmune assay, a tracer that is labeled ligand, a binder, antibody which is a specific antiserum, a separation system to separate the bound and free antigens, a standard in highly pure form, and a free human antiserum. These reagents we required in radioimmune assay. Reagent used in radioimmune assay tracer, the radioisotopes used are beta emitters, tritium and C14, carbon 14, gamma emitters, iodine 125, tritium, weak beta ray emitter, significantly lower energy than carbon 14, long physical half-life 12.3 years, biological half-life is 10 to 12 days, produced by neutron bombardment of a lower hydrogen isotope, used for the drug like protein and amino acid. That is use of tritium, carbon 14, weak beta ray emitter, long physical half-life, that is 5.7 into 10 raised to power 3 years. Biological half-life bound is 12 days, unbounded is 14 days, commercially available as barium carbonate, C40. Now, iodine 125, low gamma emitter, that is 35.4 kilo electron volt, high specific activity, short physical half-life, that is 60 days. Positron and gamma isotope. The positron beta emitting radionuclei are mainly restricted for in a vitro experiment. The gamma emitting radionuclei are useful for in vivo imaging. Other commonly used isotope positron C11, nitrogen 13, oxygen 15, fluorine 18, etc. gamma, that is emitter, that is indium 111, 111, iodine 123, iodine 131, samarium SM 153, and selenium 75. Iodine 125 is used most favored because it can be obtained with specific activity and almost 100% isotopic abundance, thus the reducing counting time and being economic. The convenient half-life, that is 60.2 days, has shelf-life for labeled antigen is long. Iodine is natural constraint of thyroxine and triiodothyronine. It can be easily introduced into a peptides molecule, steroids, gamma emission permits the use of simple, inexpensive equipments for the counting radioactivity. Reagent used for radioimmunoassay, that is disadvantages. The it damage to the ligand may occur during the storage, health hazards, and other disadvantages. Similarly, tritium is more efficient when relatively small sample is to be determined. Advantages of tritium: long shelf life, that is 12.3 years, highly affinity, and no necessity of derivatives preparation. Minimal health hazards. Disadvantages: uh, requires scintillation counter, which is costly, and low specific activity, that is as compared to carbon 14 and iodine 125. It is replaced by injecting the antigen together with fruits adjuvant into a suitable animal such as guinea pig, rabbit, or goat. Molecule like thyroid hormones or steroids drugs are not immunogenic, so they are conjugated to the carrier protein and polymers to make them immunogenic. Separation system, it's required because the bound fraction does not precipitate spontaneously at the low concentration, and variety of procedure are available. Physical method: filtration, chromatographic, electrophoresis, charcoal Dragstone adsorption, etc. Similarly, chemical methods: organic solvent such as ethanol, dioxane, polyethylene glycol (PEG), or salt such as sodium, zinc, ammonium sulfate. Third is solid phase system, a standard that is a ligand analytes in highly pure form. Drug, protein, hormones, etc. must be in pure form so they can diluted standard or prepared in ligand-free serum. In case of protein, hormones, the standard should be prepared preferably with the hormone from the species from which the serum is going to be analyzed. Ligand-free human serum. It is prepared by treating human serum with charcoal. It can also be prepared by collecting serum from volunteers in whom the production of that ligands of hormones has been inhibited by the treatment with an appropriate drug. A known quantity of antigen is made radioactive. General procedure for the performance of radioimmunoassay. A known quantity of antigen is made radioactive. This radio-labeled antigen is then mixed with known quantity or amount of antibody for the antigen. As a result, the two chemical binds to one and another. Antigen react with antibody. A sample of serum from a patient containing unknown quantity of same antigen is added. This causes the unlabeled antigen from the serum to compete with radio-labeled antigen for the antibody binding site. As the concentration of cold antigen is increased, more of it bind to the antibody. This is, you can see in this diagram, radioactive antigen labeled with red color and unlabeled antigen, that is blue color. Radio antigen combined with antibody and form a complex. Non-radio antigen display in red color and a radioactive antigen display in red color and unlabeled antigen display in blue color. This you can see the radioactive of supertant and free antigens in this diagram. Radioactivity of precipitate bound antigen is given here. All blue dots is attached with antibody and radioactive antigen also is attached with antibody. They compete with each other. Now advantage of radioimmune assay. The radioimmune assay is very sensitive technique used to measure the concentration of antigen without the need to a use of bioassay. It can measure 1 trillion 10 to 12 of a gram of material per milliliter of blood. That is very small quantity. It is structurally specific as antigen antibody reaction are highly specific. It is indirect method of analysis. It is a saturation analysis as active reagent added in a smaller quantity than that of analyte. Disadvantage of radioimmunoassay. This is prolonged reaction time in a days as consequences is highly diluted reagent is used. Radioactive iodine is used in is not a cheap reagent, that is very expensive. Possible health hazard due to handling of radioisotope may be possible. All the reagent must be added precisely, very accurately. Limited assay range, lack of direct linear relationship between analyte, concentration of signal response, difficult of automation, and lengthy counting time. Use of radioimmunoassay for the detection of narcotic drugs, blood-brain screening for the hepatitis, early cancer detection, measurement of growth hormones level, tracking of the leukemia virus, diagnosis and treatment of peptic ulcer, and research with brain chemicals called neurotransmitter, etc. This is recent applications of radioimmunoassay. We can use for in pharmacokinetic study, measurement of estradiol or estradiol translational study in breast cancer, plasma estrogen measurement, development of aromatase inhibitors for endocrine therapy, significance of prostate specific antigen in prostate cancer, patient, and in non-cancerous prostatic disease patient, evolution of enzyme immunoassay and radioimmunoassay method for the measurement of plasma oxytocin. So this is a simple revision of radioimmunoassay. You can see uh, you have to write principle of radioimmunoassay, method, procedures, reagent used in radioimmunoassay. What is the advantages? What is the disadvantages of this technique? You must or you have to write in your exam. That is an important chapter, and 7 to 14 marks question will be asked in your university examination point of view. So friend, if you got any doubt in this chapter or any topic of our subject, you can directly ask me on telephone or WhatsApp group. I will try to solve your difficulties or problem. Thank you. Thank you very much.